I'm sure that many of you have known Dr. McLaren for your entire training career. I've known Dr. McLaren for as long as I've known what eggs were, and have had the good fortune to meet her a couple of times through the years. So we're just delighted she's here. She's actually here visiting Dr. Biggers and Betsy. They spent a couple of days in New Hampshire. So we're thrilled to have her. And I've asked John to introduce her because Dr. Biggers knows her career and her goals and her future goals, although maybe you're going to tell us about that as well. One of the things I think everyone in this room is aware of is that a challenge that faces any kind of stem cell researchers, especially if you want to do what the South Koreans just did, is the ethical issues associated with asking women to donate eggs simply for research. And although we've sort of fussed with that for some time, I think we have that pretty well in hand. As a foundation, the country at large is still struggling with that. And Great Britain has sort of paved the way for years on establishing ethical standards for this kind of research. And Dr. McLaren has been a major player in that. And I'm sure that John is going to tell us a little bit about her career there. Dr. Biggers?
It's a great pleasure for me to introduce Anne. She's been a long-term friend and a former colleague of mine. I think we first started working together in 1956. Anne's had a very distinguished career, and I'm not going to begin to cover all the things, but she is a fellow of the Royal Society of London, and I think had the distinction of becoming the first woman to hold office in that august body. She was Foreign Secretary of the Royal Society for 5 years, I think. She's a former president of the, well, what I would say, she started life as a virologist, and we were fortunate she switched to reproductive biology, the developmental biology. She's been president of the British Society for Study of Fertility, and president of the developmental biology. She played a very significant role as a member of the British Warlock Committee that developed the legislation, or the White Report, I think it was, and legislation that led to the what some regard as the model laws for regulating experimentation with human embryos. And, I know she played a part testifying before the British Parliament on this subject. She's represented Britain on the ethics committee, whose name I can't remember, in Europe. She was awarded the Distinguished Japan Prize 2 or 3 years ago. So you can see she's had a very remarkable career. And I'd like to now introduce her to give her talk.
Well, thank you, John, for those kind words. And I'm very grateful to Anne. When Anne invited me to talk at this Activated Egg Symposium, I was delighted and honored, but also somewhat reluctant, because actually I don't work on eggs, and I've never worked on eggs, and I don't actually know much about eggs. So what I thought I'd do is just to go through all the reasons I could think of why we might want eggs, and then how we can get hold of them. So that's what this talk is going to be about. And I don't know how long it'll be, because I think I wasn't told until about 5 minutes ago what the program was. Let's see how it goes. Anyway, human eggs, well, first of all, how do we need them? And here is a human egg. Oh, that's funny, the human egg got lost. There's the human egg. With its first polar body and its second metaphase plate and nice cumulus cells all around it. I think that's a human egg. And I reckoned that there were 3 main areas as to why we need those eggs: the traditional, clinical, and for purposes of research. Now, of course, the traditional use of eggs doesn't really need much explanation. At the top, we have one of my favorite paintings, Cranach's Adam and Eve. And at the bottom, we have the more post-coital Botticelli of Mars and Venus. So that's all I shall say about the traditional purposes of eggs. It's obviously to make babies, and none of us would be here if it wasn't for those eggs. Then, what about clinical? Well, unfortunately, there are a number of women and couples who aren't able to make babies that easily. And so assisted reproduction, infertility treatment has become a big thing. And I think I'm right in saying that one of the major causes of infertility is problems with ovulation. But I'm not a medical, but I think I'm also right in saying that this is one of the easiest of the infertility problems to actually treat and help by hormone treatment. Of course, the dose of hormone needs to be carefully controlled, and also the ultrasound screening is very important, so there are not too many eggs shed, because there are these awful stories of unfortunate women who found themselves pregnant with, I don't know, 7, 8, 10 fetuses. Okay, but then if the ovulation problem is not everything, then obviously it's IVF or ICSI that comes next. Egg recovery from hormone stimulation, more eggs than would normally be shed, because not all are fertilized and not all the fertilized ones develop. ICSI is, of course, if the sperm doesn't do its stuff with ordinary in vitro fertilization, a single sperm can be injected into the egg, and amazingly, that seems to be very effective. At the time when it was first introduced, people were, especially the sperm people, were astonished, because there'd been an idea that, there was sperm selection and only the very best one to get there would do the fertilization. So just taking one at random and injecting it seemed very curious, but there it was. It worked. So IVF or ICSI. Then egg donation. That, of course, is an important reason for wanting eggs. Age there refers to the age of the woman, and therefore the time since that oocyte was first laid down before birth, rather than how long the egg has been out of the body. So as women age, their eggs get less and less developmentally competent, and very often with egg donation from a younger woman, the fertility is restored to a very high level. Then there are the anovulatory women who don't ovulate eggs at all, let alone the quality, and one of the commonest causes there is Turner's syndrome, having only one X chromosome instead of 2. And these women are quite fertile if they can have donor eggs. And this is one of the chief reasons in my country for the long waiting lists for donor eggs. Then the final clinical reason for wanting eggs is for women who need pre-implantation genetic diagnosis. there are some couples who are at risk for a serious genetic disease because they're both heterozygous for a gene for one of the many known serious diseases, and previously they would either have had to not have children at all or got pregnant not knowing whether the baby was going to be born with this genetic defect or not, and perhaps having either chorionic villus sampling or amniocentesis, and if the result was bad, they would then usually have a termination of pregnancy. Now, pre-implantation genetic diagnosis, as you know, avoids that. If you start with an 8-cell embryo, you can take out 1 or 2 blastomeres and use those blastomeres for usually a PCR, polymerase chain reaction, to see whether or not they carry the genetic defect in homozygous form. Here there are 5 embryos which have been sampled, and 2 of them did indeed carry the defect in homozygous form, so they obviously would not be returned to the woman's uterus, but 3 were okay. There's also the possibility of taking a few cells from the trophectoderm at the blastocyst stage, but although this is explored from a research point of view, it's not used in clinical practice. And this other side just shows that you could also do it after cryopreservation. So that's pre-implantation genetic diagnosis. Okay, so much for the clinical need for eggs. What about research? Well, I've listed a number of types of research here. First of all, we know a lot about the biochemistry and physiology of eggs, but still not enough. There are many unanswered questions, and if we knew the answers to all those questions, it would be easier to design even better than culture media for culturing embryos than already exist. And as most of you know, John Biggers has been extremely active and influential in this whole area. So if there are any questions about the biochemistry and physiology of eggs and the need for further research in that area, John's the person to ask. Then research about infertility treatment. In vitro maturation. Now, there are clinical conditions, and polycystic ovary syndrome, PCOS, is one of them, where I think too many follicles start developing, and then they get ovulated prematurely at stage 7 or stage 8. And of course, at that stage, they're not developmentally competent. They would normally become matured in the ovary in vivo before they were ovulated. But if they're ovulated prematurely, they can be matured in vitro. But again, the conditions and the culture medium, the hormone support and all that has not been very well worked out. There have been a number of pregnancies from in vitro matured eggs, which have then been either fertilized or ICSID, but it's by no means optimal. Recently, the treatment with in vitro matured oocytes has been licensed in the UK, and there have been some pregnancies. But as I say, this is an area where further research would be extremely useful. Rejuvenation of aged eggs. This is a question people have tried with these, and when I say aged eggs, again, I mean eggs from older women which have been oocytes since before birth, and which are not as developmentally competent as they should be. It's been suggested that injection of a small amount of cytoplasm from younger eggs would rejuvenate them. Now, there have been publications on this. There have been women who have had normal babies after such treatment, who'd been trying for probably years without success. On the other hand, whether or not the pregnancies were really due to the treatment is not established, and because there are possible risks, this treatment, I think, is now not encouraged in the USA, and it is not licensed in the UK. But again, it perhaps is something that needs further research. There are two other clinical areas, medical problems, which fall in the research area at present. One is mitochondrial defects, and the other is stem cell therapy. So I'd like to say a bit about both of those. Now, mitochondria I've always found fascinating. They're those little organelles in the cytoplasm. In an egg, I think you have something like 100,000 of them. And each mitochondrial DNA strand is circular. So there are 100,000 of these little circles. Each has only 16 kb of DNA, has only 37 genes, and of those genes, most of them are RNA stuff, and only 13 code for proteins. The proteins they code for are almost all enzymes involved in oxidative phosphorylation. I can never say that, phosphorylation. And of course, the mitochondria are responsible for providing energy for the cell. And they're aliens, they're invaders. It appears that back in the distant past, in the primeval soup, little bacteria hitchhiked on larger cells and provided them with more energy, while the larger cell provided, I suppose, nutrition and protection. So that, for instance, the genetic code of mitochondrial DNA is not the same as that of nuclear DNA. They really are outsiders that have become insiders. And the important thing about mitochondria is that they are all inherited from your mother. Entirely maternal inheritance, although the sperm has half a dozen or whatever little mitochondria as compared with the 100,000 in the egg, the sperm mitochondria all disappear and get degraded within a few hours of fertilization, in normal fertilization. So mitochondria, unfortunately, sometimes have mutations, just like all DNA has, and become defective. So at the top here, we have a cell with normal, fully energy competent mitochondria, but sometimes this mutation develops and the mitochondria replicate independently. They're randomly distributed in the daughter cells, so one can get quite a variety of different cells, some which are fully energy competent, others that are hopeless, and every degree in between. And these cause respiratory defects, which are responsible for a number of very serious diseases, including some which can be lethal, and they are all maternally inherited. 1 in 10,000 women have serious mitochondrial genetic defects in their mitochondria. It doesn't necessarily mean that they show any disease, because there may be only a very small proportion of defective mitochondria which don't show up phenotypically. But 1 in 5,000 are at risk of developing the disease. And the diseases are really quite varied, and nobody quite understands why they're so varied. But the respiratory failure, obviously in the heart, cardiomyopathy, liver, special senses, a lot of deafness, and also optic problems, retinitis pigmentosa, optic atrophy, have a mitochondrial genetic component, and all these other ones too, myopathy, neuropathy, thyroid, diabetes. And as I say, occasionally they can actually lead to death. So it's a serious problem. And it's very difficult to give women who have perhaps mild symptoms of one of these diseases any sort of sensible genetic counseling. And the reason for that is that, now this is mice, this is not women, but as so often we hope that humans behave like mice behave. In mice, if one has mitochondrial so-called heteroplasmy with a mutant mitochondrion around, if you look at the primordial germ cells, soon after the establishment of the germ cell lineage, which in mammals happens during gastrulation, quite early, you find that the proportion of mutant mitochondrial DNA strands is very uniform between different primordial germ cells. Now, there's something like 9, 10, 11 cell divisions between the germ cell lineage being established and when you get primary oocytes entering meiosis, after which no further cell division happens until ovulation. So during those 9 or 10 or 11 cell divisions, these mitochondrial mutations get segregated independently, with the result that if you look at primary oocytes, they're highly variable. Instead of being very uniform like the primordial germ cells, they're highly variable. Some have a lot of mutant mitochondrial DNA strands, some have very few. And since no further cell division occurs up to the time of mature oocytes, you never know whether any particular egg is going to be really badly affected, so that any baby born after fertilization of an oocyte like that is going to be in a bad way, or whether they'll show no symptoms at all, or whether they'll be perhaps slightly affected like their mother was. So this is a real problem for genetic counseling. Is there a way around this? Well, in principle, there is. Because if you had a donor egg from a normal woman, you could remove the nuclear genetic material from that egg and transfer into it the 2 pronuclei from the parent egg. And okay, you might have just a few defective mitochondria transferred along with the nuclei, but mostly you'd have normal, clean, healthy mitochondria there, and the babies would be fine. Now, this route has not been attempted, as far as I know, in any country, but Professor Doug Turnbull in Newcastle in the UK has been interested for a long time in the possibility, because he has so many patients who are suffering in this way and want advice. And he has just been licensed, after a great deal of argument and discussion, he's just been licensed to start a program on human material, a research program. He's shown in mice that it works, proof of principle. He's taken mouse eggs and transferred pronuclei into an empty egg and got babies, and they're fine, very few of the mutant mitochondria. And what he's going to do is use eggs from IVF programs that are abnormal in that they have 3 pronuclei. Now, as you know, these would never be used for clinical treatment in an IVF program, but they can be donated for research rather than simply be discarded. He's going to remove the pronuclei from both and then put 2 pronuclei into the other cytoplasm, reciprocal exchange, so that he has 2 reconstituted diploid zygotes. He'll culture those for 2 days and do a number of tests. For instance, he'll remove a blastomere for the analysis of the mitochondrial DNA to find out whether it looks normal or not. If there is, as there often is, some heteroplasmy in the donor eggs, these ones, he'll be able to tell how much of this sort of mitochondria has been transferred to this zygote and vice versa. He'll continue culturing up to the blastocyst stage to see what proportion of embryos develop to the blastocyst stage and whether they look reasonable or not. And then he'll process those blastocysts for cytogenetic, epigenetic, looking at imprinted genes to look at methylation patterns, or mitochondrial DNA analysis. And this is a 3-year program of research. He had quite a bit of argument with our licensing authority, the so-called Human Fertilization and Embryology Authority, HFEA, because our 1990 Act has a little bit in it which says changes to, quote, the genetic structure, close quotes, of the cell of an embryo is prohibited. Now, my argument would be that such a procedure doesn't change the genetic structure, it changes the genetic content in the sense of the mitochondrial DNA content. But what the 1990 Act was concerned about when it talked about genetic structure was actually transgenesis, because at that time in 1990, well, even earlier, transgenic technology was just starting up, and people were very worried that somebody might start making transgenic human embryos. But in this procedure, there is no change to any gene. It's simply replacing defective mitochondria with normal mitochondria. So if his research program shows up no evidence of any abnormalities in those developing human embryos, I think he will then apply for a very limited program of clinical treatment of women who otherwise would not probably have dared have a pregnancy, with careful monitoring and follow-up. Okay, then finally, on the research side, stem cell therapy. And I'm not going to say much about this because you all know too much about stem cell therapy. You all know that there are all those awful diseases and that there are a whole number of cell types that if one had lots of them, you could treat the diseases. You all know exactly what a stem cell is, that it can make more of itself, or it can differentiate into every other cell type you can think of. And you also all know that you can get stem cells from an adult, and adult always means anything postnatal, really, from the brain, bone marrow, satellite cells of muscle, and they're all very useful for particular purposes and will be even more useful when there's been more research. Cord blood has stem cells in it, very useful. Then early embryos, pre-implantation embryos can give rise to embryonic stem cells from the blastocyst stage, and unfertilized eggs, if they're activated parthenogenetically, as Anne Kiesling has usefully pointed out, can also be used to make human embryonic stem cells, which would then be immunologically identical with the woman who had provided the unfertilized egg. But I'll say a little bit more about that later. Anyway, back in the early days of 1998, when Jamie Thomson published the first derivation of human embryonic stem cells, this is his picture, and the top line is what he actually did, taking the isolated inner cell mass, culturing it. The bottom line is what he hoped might one day happen, that it would make kidney cells, heart cells, bone marrow cells, etc., which could be used for therapy. And things are moving on. We've probably got this far, but not, I think, really that far, though some people would disagree. Maybe that'll come up later today. Now, as far as human embryonic stem cells go, as John Biggers mentioned, one of the major problems is an ethical problem because, of course, these come from fertilized eggs, and if you believe that the moral value of an embryo from the one cell stage onwards is equal to that of a newborn baby or an adult human being, as some people do, then any research that causes that embryo to stop developing, even if it was going to die anyway, is akin to murder. I respect that view, but I don't agree with it. Most of the people in my country think that the moral value of an embryo increases as it develops first into a fetus and then into a baby. And this point of view was reflected in the UK Parliament back in 1990 when that Act was passed. In both our Houses of Parliament, there was a strong vote in favour of allowing human embryo research under very strictly regulated and controlled conditions. But also they felt that if the research project required the creation of embryos for research, that also should be allowed. And that actually has been very useful, though it's been used in only very few circumstances. This is a list of the countries that agree with the UK in allowing derivation of human embryonic stem cells from spare embryos from IVF. I should, of course, have said that Jamie Thomson got these embryos, human embryos, from IVF clinics with informed consent of the couple, of the woman who was donating these embryos for research. And in most cases, whether they are fresh or whether they have been frozen for some years, if they're not going to be used for that woman's treatment, nor donated to another woman for treatment, they would just be allowed to die. So it's these spare embryos that are now often donated for research, and that research can include the derivation of human embryonic stem cells. However, what's this got to do with eggs? Nothing much. These are embryos. These are embryos. However, if they're going to be used actually for therapy, then there is the problem of graft rejection. Because if they have been derived from an embryo donated from an IVF clinic, they will not be immunologically compatible with the patient who is being treated with the stem cells. And one possibility that has been put forward is that if one used somatic cell nuclear transfer, that is the cloning procedure, the same procedure of transferring a nucleus, a somatic diploid nucleus, into an empty egg, the same cloning procedure that was made for using Dolly the cloned sheep and all these other cloned animals now. If one used that same procedure on a human egg, that the blastocyst and the ES cells that were derived from that blastocyst, they would be immunologically compatible with the donor of the somatic nucleus. And you all know about, it's often called therapeutic cloning, which is a dreadful term because there's nothing therapeutic about the cloning itself. The stem cells may be used for therapy later, but it's an awful term. Anyway, cloning for stem cells. The patient has the disease. This rather old-fashioned doctor is doing something down here. The somatic cells are recovered and the nuclei are transferred to an empty egg, fused. The donor egg has had its own genetic material removed. The embryo is cultured, the stem cells are recovered. They're then differentiated into whatever cell type this patient requires for his particular disease. Now, I'm very skeptical myself as to whether this very labor-intensive and labor-costly procedure will ever be realistic in any healthcare system. But as you all know, the recent papers from South Korea at least have provided proof of principle. Now, as I mentioned, Anne Kiesling has suggested that you could get from a woman an egg, her own egg, which could develop pathogenetically, and then you wouldn't need any donor egg. You could get stem cells, culture those to whatever tissue type that woman required for her treatment, and that would be fine. Tough on you chaps. But there we are. Anne has a point. This procedure is much more attractive, I think, in the US than in the UK, because in the UK there's a great shortage of donor eggs, and in the US one can at least buy donor eggs, but if a woman could provide her own eggs, this in the UK might be, I think I got that the wrong way around. I said it would be more attractive in the US than the UK. It's the other way around. I think in the UK it's difficult to get donor eggs, so the parthenogenetic approach would be more attractive. In the US, you can buy donor eggs, so that would be less needed to get parthenogenetic ones. Anyhow, even if one didn't use the cloning approach, there's a number of other ways which I think are much more realistic of how to get over the graft compatibility problem. You can use sometimes the stem cells from the patient themselves. You can use genetic manipulation to get rid of the immunocompatibility antigens, perhaps. You can have a very large stem cell bank, so usually you can get quite a close match. There are ways of inducing specific immunological tolerance in the patient, and of course immunosuppressive drugs are improving all the time. On the other hand, the cloning procedure could be of enormous importance, not for therapy, in my opinion, but for research, because there are patients with very rare genetic diseases, maybe only 40, 50, 60 patients in the whole world. We know very little about those genetic diseases, but if we could make stem cell lines from skin cells from those patients, the research would be very, very helpful. And equally patients with common but genetically complex diseases, stem cell lines would make it much easier to find out the genetic basis or the non-genetic basis of that condition. And as a result of that possibility coming to light, we've now got two licenses issued in the UK for this cloning for research. One to Alison Murdoch in Newcastle, who is going to be looking at diabetes, making stem cell lines from a number of diabetic patients, and one from Ian Wilmot in Edinburgh, who is concerned with motor neuron disease, which is a very mysterious disease where we know very little about any genetic background. Okay, so that's the first part. That's why we need eggs. How can we get them? Well, possible sources of oocytes. Donated by women. And this, for clinical use, is the only practical method at the moment. Now, the problems here are commercialization, what the philosophers call commodification, exploitation of women. If you pay women large amounts of money, they may donate eggs, but there are risks to health. There are certainly risks, but not risks, but it is time-consuming and uncomfortable to have all the hormone injections needed for egg donation. There's also the risk of hyperstimulation unless the ovary is properly screened by ultrasound, and of course hyperstimulation can lead to death. There have also been suggestions that maybe there might be a risk of ovarian cancer later if there's been a lot of egg donation, but there's been no good evidence to support that possibility. But in Europe, I think not only in the UK, but also in Europe in general, it is not permitted to pay money for egg donation. In the US, it is permitted, and I believe quite large sums of money are sometimes given for donated eggs, and I've even heard that young women are able to buy their way through college by donating eggs. So that's a different ethical stance. In the UK, we do allow, and there's been a lot of discussion about this, but we do allow what we call egg sharing. Now, most IVF treatment in the UK is private and quite expensive, and some is on the NHS, but not enough. So a lot of couples can't afford IVF or can only afford one cycle. But if the woman produces a reasonable number of eggs and they fertilize well, she is permitted to donate some to another couple who needs egg donation, and then she gets a second cycle free or at a very reduced rate. Now, the argument in favor of that is that it gives two women a chance of the babies they really want, and without it, neither would have such a good chance. The argument against is that it's tantamount to buying eggs, but the ethicists argue about whether money is different from exchange in kind, and I'm not going to go into that. Fail to fertilize oocytes. These are from IVF clinics where the egg is put with sperm, 24 hours later, no pronuclei, no fertilization. Now, these eggs have been used, for instance, Roger Peterson used them in San Francisco and successfully made human embryonic stem cell lines from eggs of this sort, and it's what Alison Murdoch in Newcastle will be using for the research. But I don't think it's at all practicable, at least for clinical use. Then in vitro maturation of immature or primordial oocytes. Now, we're not talking about the same sort of in vitro maturation that I mentioned earlier. Before, we were talking about this stage 7, stage 8 in vitro maturation. What I'm talking about now is these little tiny primordial follicles with primary oocytes in them, of which there are, well, before birth, maybe hundreds of thousands in every ovary, but even after birth, tens of thousands, and every adult woman of reproductive age has large numbers of these primordial follicles in the ovary. Now, if these could be recovered, a small slice of ovary removed and matured in vitro, they could be a source of almost unlimited eggs for donation. John Eppig in Bar Harbor has done a lot of work on this in mice. He managed to mature these to a stage where they could be fertilized, but in the early experiments that he did, out of hundreds and hundreds that he tried, only one turned into a mouse. And that mouse was, for obvious reasons, called Egbert. And it was a miserable mouse. It had all sorts of defects, all sorts of medical problems. And for a while, he abandoned that form of research. But he started again recently, and I'm not quite sure what he's doing different. I don't know whether anybody quite knows what he's doing different. But anyway, he now has, I believe, dozens of very healthy, fertile, happy Egberts running around in the lab. So that's interesting. Then there's a possibility, again for research only, of using animal oocytes. Hui Zhenshen in China used rabbit oocytes, removing the rabbit genetic material, putting in human skin cells, getting good blastocysts, which were indubitably human in the sense of the nuclear DNA, making stem cell lines, but the stem cell lines didn't do all that well, and I haven't heard much from her recently. There's also been attempts, I think, to use cow oocytes in the same way, and this might be useful for research. Then, oocytes derived from embryonic stem cells. This is a fascinating development in my view. The paper, for example, from Hans Schürler's group, which reports Hubner et al. Used embryonic stem cells, which we've already talked about. Pluripotent, can make all cell types. Because they can make all cell types, it's reasonable that they should also be able to make germ cells. And sure enough, when he cultured embryonic stem cells, this is all mice, he found patches of cells eventually, which showed OCT4 oocyte specific germ cell, no, not oocyte, germ cell specific OCT4 transgene lighting up. That's these green patches here. And he also found they looked sometimes as though they were migrating, which is the time at which they show this specific OCT4 transgene. He also found cells which showed this red vasoprotein, which is expressed in post-migratory germ cells after they've reached the genital ridge. And he got these clumps of cells, rather like what you get in vivo, expressing both the OCT4 protein and the vasoprotein. He followed them for another couple of weeks and then found that some of the cells detached and started staining for a marker which is specific for meiosis in germ cells. These are derived from the ES cells. These are derived in vivo. And he showed that the SCP3 marker was expressed in the nucleus, and the nucleus started looking a rather diploidine type configuration. The pictures in his paper, of course, are much better than these. And finally, he got cells that looked like mature oocytes. They got extruded from a follicle, something that looked like a follicle. They had something that looked like a polar body. They were in a zona of sorts, but a rather fragile zona. And if these went on for another few weeks, and we're now up to, what's this, seven weeks more or less, 6 weeks, he found within these groups of somatic cells, follicle cells, he found what looked like a 2-cell embryo and eventually 3 cells. Again, you can see the zona pellucida around, 16 cells showing the specific OCT4 protein, developing into blastocyst-like objects. Some of the blastocysts looked very like proper blastocysts, and again OCT4 in the inner cell mass, and PCR analysis showed that they did indeed express all the right genetic markers. Now, these actually look extremely like the sort of pictures that one gets from parthenogenetic development of the LT strain of mice. So maybe they have something in common with that. We don't know. We don't know whether they're developmentally competent. These blastocysts were not put back in a uterus as far as we know, so that's that. But there are other labs also which have pursued this line. This is a diagram from as in Sarani's News and Views. For example, in Daly's lab, Gayson et al., they made embryoid bodies from the ES cells and selected for treated with retinoic acid, which makes primordial germ cells multiply. They found that some of the somatic cells were expressing the SRY gene, which is the testis, specific testis gene, and within those patches of SRY gene, they got the germ cells becoming apparently haploid and turning into what looked very like sperm, which were then injected into eggs to fertilize those eggs. But again, the blastocysts that resulted from that fertilization, as far as we know, have not got any further in development. Torioka et al. In Japan, that's in Nose's group, they also got male germ cell development from ES cells and put them with testis, somatic tissue, and got sperm. And in Australia, they've done the same sort of thing with human embryonic stem cells and got apparently primordial germ cells and embryoid bodies. So the final possibility before I get onto that, oocytes from bone marrow or peripheral blood. And this possibility was raised, first of all, in a paper from Jonathan Tilley's lab, Johnson et al., last year, where they showed pictures of what they thought were oogenic stem cells in an adult ovary. The pictures in that paper were not terribly convincing. To me, they looked rather like what you get in cultured ovaries when the oocytes are actually exiting from the edge of the ovary. And I wasn't convinced that they were really undergoing mitosis and dividing, and it wasn't clear whether any of them were undergoing meiosis to develop into oocytes. This year, he has another paper from the same group, now in Cell, showing that in infertile female mice, whether they were genetically sterile or chemically sterilized, that you could apparently get new oocytes appearing if you treated with bone marrow transplantation or even peripheral blood. And he claimed that these adult mouse ovaries could produce hundreds of new oocytes within 24 hours. Now, that in itself is a surprising claim, because normally to get from oogonia to meiotic oocytes takes at least a week. And one couldn't help wondering whether there were pre-existing follicles that perhaps after the chemical treatment then started maturing again. I don't know. But he used donor-derived GFP-labeled oocytes, and they seemed to turn up in the ovaries after the peripheral blood transplantation. And the pictures from the bone marrow transplants looked particularly convincing, more convincing than the peripheral blood transplants. Now, if this is really true, I think it's fascinating.